Today, I will be introducing our assigned topic, which is the AgroBacterium species. This is a very interesting group of soil bacteria because they don't just infect plants, they actually have the ability to transfer their DNA into plant cells. Because of that, AgroBacterium is known both as a plant pathogen and a useful tool in biotechnology. Researchers use an agrobacterium as a tool to create genetically modified plants, improve crop traits, and study plant genes. In short, agrobacterium is a both challenge in agriculture and a powerful tool of biotechnology. So in this presentation, we will explore what agrobacterium is, how it causes diseases, and why scientists use it for developing improved crops. So for introduction, Agrobacterium sp is a type of soil bacteria known for transferring genes to plants. This natural process of gene transfer has made Agrobacterium an important tool in plant biotechnology. These bacteria typically infect plants through wounds that causes plant diseases like crown gall. However, scientists have utilized these natural mechanisms for beneficial purposes, introducing desired traits like pest resistance, improved yield, and environmental tolerance in plants. Studying agrobacterium help us understand how bacteria interact with plants and how this process can benefit agriculture.
Good day, everyone. My name is Boots Tokbo, and now let's talk about the history of Agrobacterium. In 1897, Frigiano Cavara observed bacteria causing galls on grapevines, including tumors. Cavara's work was foundational in identifying a bacterial cause for plant tumors, which later became known as crown gall disease. In 1907, Erwin Smith and Townsend officially named it Bacterium tumefaciens, confirmed as cause of crown gall disease. Smith and Townsend showed that this bacterium entered plants through wounds and caused uncontrolled cell growth similar to cancer. In 1940s, Armin Braun introduced the idea of a tumor introducing principle or TAP transferred by bacteria to plant cells. Braun proposed that the tumor-causing factor was a stable substance produced by the bacteria which altered the plant cell behavior even when bacteria were no longer present. While in 1970s, discovered that TIP is a DNA segment from the bacterium plasmid incorporated into plant genomes. Identification of the plasmids tDNA as the transfer genetic material revolutionized understanding of bacterial-plant interaction and genetic engineering. In 1980s to present, use in genetic engineering to insert genes into crops for improvement in research. Simplified binary vector system were created to efficiently transfer desired genes into plants, enabling major advances in crop science. Now in 2001, taxonomic reclassification proposed merging Agrobacterium into Rhizobium, controversial and mixed usage. Despite the proposal, the classification remains debated among scientists, reflecting complex genetic relationship revealed by molecular data. Now, in recent updates, genomic data resulted in species reclassification, maintaining in some Agrobacterium and other in different genera. Modern phylogenomic studies have refined species boundaries within Agrobacterium, leading to recognition of new lineage and some resigned to other genera.
A pleasant morning to everyone. I am Charles Dexter Tunasan and I will talk about the uses of agrobacterium. With that, let's start. Now, the agrobacterium sp is a type of soil bacteria known for transferring genes to plant. This natural process of gene transfer has made agrobacterium an important tool in plant biotechnology. These bacteria typically infect plant through wounds that cause plant disease like crown gall disease. Now, here are some examples of the crop that are being affected by agrobacterium. As you can see in the picture, the grapevine is being affected by the agrobacterium as well as the apple. And the next slide are the walnut tree being affected by the crown gall disease as well as the rose. The question is, what is the mechanism of agrobacterium? Now let's talk about it. The production of agrosine 84, a bacteriocene that specifically inhibits pathogenic A or Agrobacterium tumefaciens strain by interfering with their plasmid replication. Now, it colonized wound site rapidly, preventing pathogenic strain from establishing infection. As a genetically modified strain, K1026 was developed to prevent horizontal transfer of the agrosine plasmid to a pathogenic strain, enhancing biosafety. Row.
Today, I will discuss the current trend of Agrobacterium sp. The study of Rosario Stuti and Yudha on year 2022, entitled Potential of Rhizobium sp. And Agrobacterium sp. And Formation of Root Nodules and Stimulating Growth of Arachis hypogaea L. This study inoculated peanut plants with the Rhizobium sp. And Agrobacterium sp. Using different carrier materials, such as peat mixed with mokuff, manure, or Loria Bertani liquid. These carrier combinations acted as biofertilizer, supporting the survival and effectiveness of the bacteria. There are 12 treatments, but let's focus only to the treatments na agrobacterium, the P2, 5, 8, and 11. Table 1 shows plant growth results. Compared with the uninoculated control, the P0, all agrobacterium treatments shown strong improvements. Just look at the P2, 5, 8, and 11. Although all contained agrobacterium sp, their results differed because the carrier materials affected the bacteria's survival and efficiency. Overall, treatments with carriers, especially the P5 and 8, give the best growth and nodulation, showing that suitable carrier materials enhance agrobacterium performance. This study concluded that Agrobacterium sp bacteria can be used as an alternative to Rhizobium, since they are closely related because they belong to the same taxonomic family, the Rhizobiaceae, and they also have similar DNA sequences. This slide shows the treatment description of this study. You can just read this one. Advantages and disadvantages of Agrobacterium. Let's discuss first its advantages. First, its natural gene transfer mechanism. Basically, the bacterium already knows how to transfer DNA into plants on its own. We're just hijacking its natural system to our advantage. Next, it works on many plants. It's not picky. It can infect a broad range of plant species, from tomatoes and tobacco to important crops like rice and corn. It can also deliver large genes. Unlike some other methods, Agrobacterium is great at transferring really big chunks of DNA, which gives us more flexibility. We can also grow whole plants from it. After we transform a single plant cell, we can regenerate an entire new plant from it that carries the new trait. Last is its simplicity and cost effectiveness. Overall, it's a relatively straightforward and cheap technique compared to more complex lab methods, making it a popular choice. So because it's natural, versatile, and efficient, Agrobacterium has become a fundamental tool for modifying plants.
For this advantage, despite its many benefits, the use of agrobacterium also presents several limitations. Number one, limited host specificity. Agrobacterium naturally infect dicots and only a few monocots. It is less effective in cereals like wheat and maize unless the bacterium or host plants are genetically modified. This restricts its application in some major food crops. Second is potential for unintended gene transfer. This is a risk that Agrobacterium could transfer genes to non-target organisms such as soil microbes. This raises biosafety and environmental concerns that require careful monitoring and regulations. Third is variable transformation efficiency. The success rate of transformation of varieties depending on plant species, genotype, and cultural conditions. Researchers often need to optimize protocols for each plant, which can be time consuming and inconsistent. Number four is biosafety rest. Because it is a living bacterium, Agrobacterium carries a rest of horizontal gene transfers in the environment. This potential ecological impact necessitates strict containment and handling procedures in laboratory. And for the last is complexity in handling. Finally, agrobacterium requires aseptic conditions and can be sensitive to environmental factors and antibiotics. Poor handling can easily lead to contamination or failed experiments, especially for beginners.
So, therefore, Agrobacterium sp is a type of soil bacterium renowned for its ability to infect and induce tumor. So, it is a gram-negative rod-shaped organism that is commonly found in a soil environments. Agrobacterium play a crucial role in natural plant diseases, but also has been harnessed as a powerful tool in biotechnology for a transferring a genetic material, and it's beyond nature. Agrobacterium sp transfers capabilities that have been adapted to develop genetically modified crops with a improved traits such as pest resistance and drought tolerance for a higher yield. So its role in agriculture and biotechnology continues to grow, contributing significantly to advance in crop improvement and sustainable agriculture worldwide. So overall, Agrobacterium sp is a both natural and pathogen and a vital tool in genetic engineering. With this, it presents in-soil ability to transfer genetic material, making it a central figure in biotechnology research and application.